Okay. So, can you guys hear me? Move my mask. If you're scared of me, I just negative. Okay. Okay. So, and just I guess in the Zoom online class. Okay. So, here we go. So this is my name. I'm a professor in the Department of Pharmacology, Physiology, and Science. And
Professor, we can hear you.
already on. Can you guys hear me?
Yes, good now.
Thank you, thank you. So the microphone fell, so very, very technical solution. Okay, so back to the beginning. So this is my name. So I'm going to be lecturing to you in the next 5.5 hours of fundamentals on gastrointestinal physiology. So I think you've had respiratory physiology, right, already? So when you talk about this branch of physiology, you dealt mainly with the lungs, a little bit about the heart, but mainly with the lungs. So, and then you will have, after my series of lectures, you will have renal physiology. And there, you will mainly deal with the kidney. The major problem with GI physiology, and it's not really a problem per se, it's a learning process, is the fact that we actually integrate the functions of a numerous organ systems in the GI tract. We begin in the oral cavity, we go to the esophagus, stomach, pancreas, liver, gallbladder, which is which I will show you in the next slide. And then I need to remind you that there's also going to be a review this coming Friday before your quiz, March 18th, 1 to 2 p.m., more or less. So this is what I was alluding to. So this is, in summary, GI physiology, which is really the story of food going from your oral cavity or your mouth, going through your esophagus, your stomach, and into the small intestine, then large intestine, and eventually out the anus. And, conceptually, sometimes, okay, so we know that from maybe high school biology or, undergraduate biology, the stomach secretes acid, and the associated organ that we have here is the liver and the pancreas. The pancreas is a major organ system, and it secretes bicarbonate. So how can two very, very, oppositely functional, in terms of major secretions, why do they belong in the same system? And so that really is the process of us understanding how food is digested and absorbed. And that's really the gastrointestinal physiology for you. And it's a story of how essentially what food you get how that is processed by these numerous organ systems in the GI tract. And if there is any major slide that actually can summarize GI physiology, it's really this slide. So understanding GI physiology is like understanding our own nutrition. And there's two general processes involved. The first major one is secretion, and the second one is absorption. So this is really a summary, I would say, chemical, reaction that's occurring. So we eat food, our oral cavity take in food, and then to that we add mainly enzymes, so we can digest the food. But enzymes cannot enter the intestine really needs to work with water. Okay, these are aqueous enzymes. It needs to have water around it, but you cannot actually secrete water from your blood side to the lumen or the inside of your GI tract.
No, go. Go see Marcus. Go. Marcus will go. Marcus.
Okay, so we have Russians in the lecture. Okay, so anyway, so we have water getting into the lumen, and they need to be secreted from the blood side into the luminal side. And with these reactions, then you yield then nutrients, ions, and water. Okay, this is the outcome of the reactions that are occurring here. This is what happens. And so after this series of reactions here, you have this series of reactions, and the nutrients are then absorbed. And you don't want to lose, your GI system, they actually secrete about 8 to 10 liters of fluid every day to process your food. And you don't drink enough fluid to compensate for those ions and water. Okay, so basically you would like to reabsorb the ions and water, and that then will yield ATP for you to live your life. And of course, you will have byproducts that you will eliminate when you go to the bathroom. So you have secretion and absorption. And these two processes actually is like a transport system. We transport a lot of these water, ions, enzymes, nutrients back and forth, especially for water and ions, between the lumen and the blood side. So enzymes and nutrients are always transported with water and ions. Okay, so that's one of the things you need to remember. And these enzymes that we use to digest food are actually hydrolytic enzymes. So this is a general reaction of almost all enzymes, that we are going to be talking about. So these are hydrolytic enzymes, and these are enzymes that require an aqueous environment. So they need water, right? That's why we have water and ions being secreted into the lumen. And for macronutrients, for food, we know that carbohydrates and proteins are hydrophilic nutrients. In other words, they are composed of molecules that are relatively hydrophilic, and so they can exist in the aqueous medium that we secrete into the intestine, into the GI tract. In contrast, the major macronutrient lipids, they're hydrophobic. And you will realize from the next several hours that I will be lecturing that lipid nutrients, the lipid component of your food, actually require very special handling by the GI system. So we actually begin with food, and eventually we harvest nutrients. So this one is like the initial component that we are going to eat, and from there we extract nutrients. So the GI system is analogous to a mining operation. And from here, we, as I mentioned, we extract nutrients from, our food, chunks of raw material. So this is really the story of the GI system. Secretion, absorption, so we can extract precious nutrients from chunks of raw material. And if you have general objectives for this course, therefore it's actually very, very simple. We need to understand the contribution of those various organ systems. I will make this available to you in the GI tract to the human nutrition. So that's really this objective number one. Objective number two is how food is processed. So your nutrients can be digested and absorbed. Very, very simple objectives. And then explain the role of the anatomical portion of the GI tract. What do they do in terms of secretion and absorption? So we go back to that mining operation to give you a picture of what the GI system is like. So this is like biomedical engineering. If you're going to design a system to extract nutrients from apple, from meat, from ice cream, then in general, you begin with mechanical digestion, and that's represented here. So these are your teeth and the contractions of your stomach. Then, because this raw material can be very dirty, we autoclave it. So you have stomach acid to autoclave it. Then we have all those enzymes. So then we have reaction vessels by which enzymes from the stomach, from the oral cavity, the salivary glands, the pancreas, the liver, and all of these enzymes are then in these reaction vessels so they can liberate the nutrients. And then once the nutrients are liberated, we need to absorb them. And so we have an absorptive surface represented by the small intestine. And in order to go from the anterior to the more posterior regions, you need a conveyor belt, right? So if you can imagine a factory going from one building to the other, you need a conveyor belt, and that's really represented by the smooth muscle. Then if something goes wrong, or the first shift of workers goes to be replaced by the second shift, you turn things off, you turn things on, and that is regulated by nerves and hormones. So we do have an exquisite system of this mining operation. And then this actually elaborate system, we also have a waste processing system represented by your large intestine. And then we even have a quality control. If we eat bad food and then we taste it, this is terrible. This is 3 months old in the refrigerator. You can spit it out. But if it's 3 months old and it tastes very good, then you might take it. But then your intestinal receptors detect something really bad, and so it would initiate the process of vomiting. So it's very, very elaborate quality control. If it has gone past the area where you can vomit, then the receptors for these bad compounds in the intestine, then they then initiate diarrhea. Okay, so you get rid of bad things, and then you begin to get good things again, so you can obtain your nutrients. So if you can just think of it that way. So it's hard actually to study GI physiology in terms of organ systems, individual organ systems. You do need to listen to the entire 5.5-hour lecture before you can really begin to have a clearer understanding of what the role is of each organ system. So there are 4. So this is a cross section of your GI tract. This is the lumen or food side, and this is the blood side or the serosal side. So things are secreted from blood to lumen, and things are absorbed from lumen to blood side. And so we have this innermost layer facing the food side, and that's called the mucosal layer. It's comprised of these three sublayers: the epithelial layer, the lamina propria, where many immune cells reside, and a thin layer of smooth muscle cells called muscularis mucosae. Then we have the submucosa, and this is where the major nerves and blood vessels are located. Then we have the conveyor belt. We have the inner circular muscle and the outer longitudinal muscle, and they're there to process, help in the mechanical digestion and in conveying food from one side of the GI tract to the next. And then we have the serosal, which is mainly the connective tissue. So let's describe the, initially, let me introduce each layer. The mucosal epithelia is really where most of the action is. They're responsible for almost all secretion processes. So they make secretory products. They're involved in digestion and protecting the GI tract. They have receptors mainly in the basolateral side for endocrine signals. Basolateral side for endocrine signals that would initiate the process of secretion. They also do the work of absorbing nutrients. Different organ systems have different specialized epithelial cells, okay, but it's still the same layer. So the mucosal epithelia, for example, in the small intestine, they absorb ions, water, your macronutrients, we talked about minerals and vitamins. In general, there is no hormones that initiate the absorption. Your intestines just recognize by nutrient sensors that there are nutrients there to be absorbed. And otherwise, absorption is still generally regulated. And towards the end of your lecture, you have, I will introduce the gut microbiota, and the mucosal epithelia, as well as the lamina propria, the intestinal, the immune cells, they have receptors that sense microbes and initiate a response to those microbes. We talked about the submucosa and the fact that they are the conveyor belt. There's also capillaries there that bring oxygen in, other nutrients into the GI tract, into your epithelial cells. And then they also are tasked, these capillaries, in absorbing these nutrients and bringing them to the liver. Then we have two major categories in section 3. First one are your smooth muscle cells, which represent the contractile tissues of the GI tract, and they're found from the pharynx, the upper third of the esophagus. So the smooth muscle cells are found throughout the GI tract, with the exception of these few areas, which has skeletal muscle: pharynx, upper third of the esophagus, and the external anal sphincter. And I have already alluded to the function of the smooth muscle cells. And so and then we have the second category, we have the autonomic nervous system in the GI tract, located within the smooth muscle layer. And so this is your GI system again, from the esophagus to the anus and the rectum. And this is your spinal cord. And the parasympathetic nervous system innervates the GI tract primarily via the vagus nerve. And the vagus nerve innervates the esophagus all the way down to the middle of your colon.
Professor, if you have a pointer, it will be more great to visualize more better, because we can't see your pointer.
You cannot see my okay, let me change my pointer to I'm going to right click, pointer options, laser. Okay, can you see this better? Guys online?
Yes,
Okay, okay.
thank you.
Okay, so so the transmitter of the parasympathetic nervous system is acetylcholine, as you know already. And so we have the vagus nerve innervating everything from there to there, and then the pelvic nerves are responsible for mainly innervating the lower part of the colon, including the rectum. The PNS typically stimulate the motor and secretory activity of the GI tract. Then the second part is the sympathetic nervous system. As you know, they always work together. One opposes the other. So this is the thoracic lumbar part of your spinal cord, and these are where the ganglia that innervate the GI tract with sympathetic nerves. And these nerves, their transmitter is norepinephrine, and they usually inhibit the motor and secretory activities of the GI tract. And so these are the three major ganglia innervating the GI tract for the sympathetic nervous system. And the serosa is mainly connective tissue. They keep these organ systems in their appropriate place in the abdominal cavity. So first, let's talk about the first part then of GI physiology, which is secretion. And I will introduce what the secretions are, and then secretions typically involve the oral cavity, the stomach, the pancreas, and the liver. And they're required for enzymatic digestion of nutrients and to have provide an aqueous environment for digestion. So here, some technical, okay. Okay, so the average human would drink about 2 liters of fluid a day. And then our saliva secretes about 1.5 liters. Our stomach, actually, they secrete 2.5 liters. The pancreas and the liver and the gallbladder, they contribute 0.5 liter and 1.5 here. Even your intestines secrete fluid. So all in all, including the amount of fluid that you drink, you more or less take in about 10 liters, and so you need to reabsorb most of this fluid. Otherwise, you will die of dehydration. And at the end of the day, normal individuals, your stool, when you defecate, should only contain about 1% of what you actually secrete and drink, about 100 ml. So we begin with the oral cavity, which is responsible for secreting saliva. So it's between 1 to 2 liters per day. And that represents about 1/3 of your plasma volume, or less than 1/10 of your extracellular fluid. And most of their secretions are secreted in the proximal regions and then reabsorbed in the distal regions. Vegetarians tend to secrete more saliva. So these are the functions of your saliva. They lubricate food. They also have bactericidal properties. They have enzymes. They maintain the pH, and they lubricate the oral cavity. So, for example, help me in speaking. So these are the functions of saliva. So what is saliva then, ladies and gentlemen? Well, there's two types. There's two categories. The aqueous component, these are mainly your electrolytes that's found in saliva. These are actually very important electrolytes. I'll show you why later, especially bicarbonate. And then we also have the organic component. And that means the enzymes, for example, that are found in saliva. And these are the organic component. So, but first, let's focus on the aqueous component. So the x-axis in this graph is the rate of flow of saliva. So this depends on your salivation. Okay, so if you are eating, for example, you will be in this part of the graph. If you are actually sleeping, you will be in this part of the graph. So the x-axis is flow of saliva, and the y-axis is the concentration of ions in saliva. So you can see here that from low flow to high flow, sodium concentrations increase, bicarbonate concentrations increase with flow of saliva, chloride concentrations increase, and there is a decrease in potassium concentrations when the flow is low, and then it becomes stable. Potassium concentrations reaches steady state in the mid to high flow rates. The important part of these secretions is bicarbonate. For those of you who are in the Dental Scholars Program, or for just general information, bicarbonate secretions regulate the pH of saliva. You need alkaline pH in your oral cavity because an acidic pH actually dissolves your teeth. Right? So you don't want that. So since the bicarbonate concentration in saliva varies with flow rate, then from your inorganic chemistry, the pH of saliva would then also vary with flow rate. So that actually people with low flow rates of saliva, they tend to have bad teeth. So that would be examined further if you happen to enter the dental curriculum and be in my GI physiology for dental students. Okay, so let's dissect the ion composition of saliva as a relative to that of plasma. So again, this is the flow of saliva as a function, and the x-axis, y-axis is concentration of ions. So we talked about sodium concentration increasing in saliva with flow rate. But this increase is actually always hypotonic to that in the plasma. The sodium concentration in the plasma is about 145, chloride is 100, bicarbonate is 30, and potassium is about 5. So you can actually see that the bicarbonate concentration in saliva is higher than that of plasma, especially when flow rates are higher. So to maintain healthy teeth, you actually need to maintain a relatively high flow rate of saliva. If your salivary glands are damaged for whatever reason, if you know of people or patients who have cancers in the head area and they are subjected to radiation, the salivary glands are very sensitive to radiation, and they actually die. Some of the cells die, and so therefore the flow rate becomes really low, and the pH in the oral cavity is low because bicarbonate concentrations are low. The other ion that is higher than that of plasma is potassium. Goes from 25 to more or less a little bit less than 20, and it's very low in the plasma. So now let's switch to the organic composition of saliva. There's, of course, numerous kinds. We have proline-rich proteins. These are various proteins that have various functions, from antimicrobial to lubrication, like mucus, for example, enamel maintenance, we have, as I alluded to you, there is going to be some secretions of saliva that is bactericidal in nature and would kill bacteria and fungi. Saliva has mucus. It secretes kallikrein, which is a vasodilator. And also enzymes. So we have alpha amylase, which can digest starch. And since you can live without salivary alpha amylase, one wonders whether the function of this alpha amylase is to taste the food that you eat. Because actually, if you chew your carbohydrates longer and you let your alpha amylase work on the bolus of food that you're chewing in your mouth, over time, that food actually becomes sweeter because the amylase releases glucose into into solution, and then you can taste, your taste buds can then taste the glucose. And then you also have lingual lipase. As the name implies, this is an enzyme that can digest lipids, hydrolyzes triglycerides, and it's stable. Its activity continues in the stomach, and it's specialized in that it can catalyze and hydrolyze lipids without bile salts. I will be talking about bile salts later in lecture today. However, just like alpha amylase, lingual lipase is not really important in terms of life because you can survive without lingual lipase, except when you have genetic diseases like cystic fibrosis, or your pancreas is not functioning well. So how do you control secretion of your saliva? So here we talk about the parasympathetic nervous system. So these are activities that stimulate salivation. These are activities that inhibit salivation. So when you smell food, if your conditioned reflex, what this means actually is if after the lecture you're going to go to the cafeteria in order to get some snacks, by the time you get out of this door, your brain will interpret that decision and you will start salivating. It's a conditioned reflex. And so when you do that, then there's a group of neurons in your medulla called salivatory nucleus, nuclei for plural, and then they actually bring the parasympathetic signals via cranial nerve number 9 and number 7, and branches of these nerves would innervate the salivary gland, release acetylcholine, which binds to your muscarinic receptors, and releasing inositol triphosphate and calcium. This is your signaling lecture here. And when this happens, then you secrete more saliva. Myoepithelial cells are small, smooth muscle cells that are found on your salivary glands, and they actually help in the contractions so that they squeeze the contents of your salivary glands and release them into the oral cavity. Your parasympathetic nervous system here also actually stimulate the salivary glands, but its impact is actually minor. The major regulator of your salivary gland is really the parasympathetic nervous system. So if you want to salivate, you will stimulate the parasympathetic nervous system. If you want to cease salivation, for example, if you're dehydrated, If you're sleeping, you should not salivate. That's a grounds for divorce, right? So then you inhibit salivation when you're sleeping or when you're tired, and then these actions here will cease. So this is the main regulator of the parasympathetic system. It's the main regulator of your salivary glands. And then we have, then from the oral cavity, we have the stomach. Okay, so we now shift to the stomach, and these are the functions of your stomach. They act as the autoclave. They also digest mechanically. When you eat, the movement in your stomach is due to its efforts to digest mechanically. They secrete an enzyme that hydrolyzes proteins. And I wanted to get to here, food storage. Okay, so we have, the stomach is not like a static box that, okay, when you eat, okay, so then you put food and then the stomach stores food. Unluckily for you, for your waistline, the stomach likes food as much as you do. So when the chemoreceptors in your stomach senses food, then it magically expands the box and say, lo and behold, you can eat more because now I'm going to expand, to the chagrin of your waistline, of course. It secretes the intrinsic factor, and then they are also involved primarily in quality control, which is vomiting. The stomach. So this is, your stomach. This is your esophagus. This is your cross section of your stomach. This is your duodenum, the proximal region of your intestine. And so these are the parts. Okay, so this is the cardiac part here, that your physicians will tell you. This is the top part, is a fundus part. And then the middle part is a corpus, or the body of your stomach. And this is the pyloric sphincter here, right below the antrum. So sometimes some books call this the pyloric region, or this is, so this is the more distal part of the stomach. This is the more proximal part of the stomach. And if you were to cut the stomach across, and then you look at it, you will actually see that it has holes in it. It's like invaginations going into the wall. Okay, those are called gastric pits. And those are actually the gastric glands. And the cell population of those glands depend on where the pit is located. Okay. So if it's located in the fundus region, so this is a cross-section of the stomach. There is a cross-section of a gastric gland found in this upper region of your stomach, in the fundus region. So the top cells are surface mucous cells. Their primary secretion is mucus and bicarbonate, and I will show them to you a number of times. Below that are mucous neck cells, and mucous neck cells, as the name implies, also secrete mucus, but they are also thought to be the stem cells in the gastric glands. So these are proliferative cells. When they migrate upwards, they differentiate into mucous cells. When they migrate downwards, they tend to differentiate into these other types of gastric cells, namely parietal cells. Parietal cells are these crocodile-looking cells here. They are responsible for secreting acid, intrinsic factor, which I will tell you what this is about, but This is necessary for cobalamin, vitamin B12 absorption. And then enterochromaffin cells, or ECL cells, are they responsible for secreting histamine. Chief cells are responsible for secreting pepsin, or when they are secreted as pepsinogen, because there's never any active pepsin in these cells. Remember, these cells are also meat. Okay, as far as the acid of your stomach is concerned, as far as the pepsin of your chief cells are concerned, they don't really care whether they're digesting a steak or the wall of your stomach. They don't. They can't distinguish those two. So that's why you need to regulate their secretion very well. And that's why chief cells secrete them as inactive precursors called pepsinogen. And when they're activated in the lumen, they are then called pepsin. If we take a gastric pit from the lower part of your stomach, this is the gastric gland that we will see. So food side, blood side, excuse me. So we still have your surface mucous cells secreting mucus, shown here by this brown arrow here that's meandering through the gland. Mucous neck cells, the same function. Then mucous neck cells migrating down, they differentiate into D cells, D as in David, cells. The function of D cells is to make somatostatin, which is secreted into the blood side. And somatostatin is a paracrine, mainly, sometimes endocrine secretion, primarily paracrine. And I will describe what its function in the next couple of cells down the road. G cells are also found in gastric pits, found in this area. And G cells make gastrin. The hormone gastrin is made by G cells, and G cells secrete gastrin into the blood side. So, ladies and gentlemen, when you study GI, make sure that you label what side is food side, what side is blood side, because you can secrete. Okay, here your surface mucous cells are secreting mucus into the food side. Your ECL cells are secreting histamine into the blood side. Chief cells are secreting enzymes. Where would these be secreted? You know by the name that you don't want pepsin to be going to the blood side, because then it will eat away your stomach. So this is chief cells are secreting their products, secretory products, into the food side. Okay, so now let's go and look at the secretions. So we have an aqueous component again, and an organic, numerous organic components. What are these organic components? We alluded to the pepsinogen secretion by chief cells. They're also called peptic cells. And because I need to tell you both these names, because when you go and look at books or and internet information, both these names prevail, and they're one and the same cells. And be careful when you're surfing the internet, because they're not Dr. Ferraris. Dr. Ferraris didn't write them. I've been teaching this for over 20 years, so I know what's right and what's wrong. So not all of them are correct. Okay, some of them are old, so you need to be aware of that. So pepsinogen, at pH less than 5, the pepsinogen is converted to the active form pepsin, and which then catalyzes its own conversion. And the activity of this enzyme, proteolytic activity, is greatest in the pH 1 to 3. The second is intrinsic factor secreted by parietal cells. So if in the more modern world of medicine, bariatric surgery is very popular. Do you know what bariatric surgery is? Okay, so you staple or even remove part of your stomach. So if you have a complete removal of your stomach, you can live with one adaptation. You need to be injected with vitamin B12. Okay, because you need vitamin B12 to make your red blood cells. And this is the only, if you have your stomach intact, then you have intrinsic factor, which helps you absorb cobalamin, which is found in your food, meat, etc., etc. And you can have a normal life. But for those who undergo bariatric surgery or have stomach cancer and removal of the stomach, you need to be supplemented with cobalamin. Then the mucus is, of course, a very important component of organic component of gastric secretions. So now let's switch to aqueous secretions. Y-axis, ion concentrations, x-axis, rate of secretion. So this theme is very, very popular and consistently repeated in the various regions in your GI tract. When your GI tract, when you're sleeping, again, your GI tract doesn't function, right? So it has to regulate its secretions because the pH of the secretion of your stomach is very low when it's active, when you're eating steak, right? But when you're sleeping, you don't want to be digesting your stomach. So you want to turn things off because, as I mentioned, your stomach acid and your pepsins don't care whether it's digesting the steak or digesting and making a hole in your stomach wall. So basal secretion when you are not eating, listening to my excellent lectures, then you have sodium and chloride as the primary secretion. Okay, so this is it when you're having fun learning GI physiology, right? The best physiology of all. Okay, so so you could have, and then I need you to know, because you will actually know that, need to know that what is the concentration of ions in the plasma. It's very easy. Sodium 145, chloride 100, bicarbonate 30, potassium 5. Those are your four major electrolytes. So you have your sodium chloride here. Your sodium is less than plasma because it's 145 in plasma. Chloride is greater than plasma because it's 100 in plasma. Okay, so all those questions I can ask easily. So this is then your basal secretion, and then you go and reward yourself for learning GI physiology, and you stimulate and reward yourself with the ice cream in the cafeteria, and and nuts and donuts, that whatever they have there, and you secrete hydrochloric acid. And this is the stimulated portion. So the flow rate increases. When the flow rate increases, the composition of gastric juice, just like the composition of salivary secretions, change. That is a constant, that is a paradigm. That's life of GI physiology. When flow rates are changing, the composition of the secretions also change. Okay. So, and now instead of sodium chloride, when you're listening to my lectures, then you have hydrochloric acid when you are actively eating. And then not only do you have hydrochloric acid, you also have a lot of this acid now in your gastric juice. And your proton levels in your, proton secretion is much greater than plasma. So here, proton concentrations are much greater than plasma. So why is that? So if you're actively secreting hydrochloric acid, then if you look at the lumen of your stomach, then it's full of ions. The pH is about 1. The concentration in millimolar, or more accurately, milliequivalents per liter, is 140. So that's a ton of protons over there. If you look at your gastric circulation or your cytosol, the pH there is 7.4 and very low concentrations of hydrogen ions. So there is an enormous transepithelial gradient from blood to lumen of chemical gradient from blood side or cytosol side to the lumen side. It's about 1,000,000 to 1 gradient. You just need to divide that by this, so 1,000,000 to 1 gradient. So what actually happens when you're moving chemicals uphill? You need to spend a lot of energy. So your stomach actually consumes a lot of ATP when you're actively eating. The transepithelial electrical gradient is modestly favorable, but it's not really sufficient to offset this uphill chemical gradient. So again, there is an enormous chemical gradient from blood side to lumen side, or from cytosol to food side. And this is your parietal cell, ladies and gentlemen. So this is the food side, this is your blood side. So this is where your steak is. So how does a parietal cell secrete protons? Okay, so we begin with the simple ingredients, water and CO2. I mentioned to you that the parietal cell is a very metabolically active cell. So in other words, it's making a lot of CO2. So this waste product is being taken advantage of by the parietal cell. It's acted on by its carbonic anhydrase, okay, Ca2, carbonic anhydrase, making carbonic acid and eventually having protons and bicarbonate as its final products. The proton is then secreted into the food side from the cytosol against that enormous concentration gradient and in exchange for potassium. So this is why it's called proton potassium ATPase, proton potassium exchanger. To maintain electroneutrality, because this is, you are actually recycling this potassium via this potassium channel, you need to secrete chloride as well. So this potassium channel here is to actually circulate your potassium. And the net secretion is really your protons here. That's on the food side. On the basolateral side, we have this chloride bicarbonate exchanger. So the protons go to the food side and the bicarbonates go to the blood side. When there is a lot of acid being produced by your stomach, by the parietal cell, this also means that your parietal cell is making a lot of bicarbonate. So much so that the pH, when you're actively eating, the pH that is leaving the capillaries that are taking blood away from your stomach to the main circulation, the pH there is actually significantly higher than systemic pH. It's because of this activity of this and this, mainly this one, extruding a lot of bicarbonate to the blood side. Of course, we have the sodium potassium ATPase, and then we also have the sodium hydrogen exchanger playing a minor role in electrolyte balance. Okay, so the question is, what is basolateral? So this is your parietal cell. So this is your apical membrane, and beside this parietal cell is another cell. You can imagine another parietal cell here, or another cell type, but their connection is actually very tight. So that from here, the membrane, the plasma membrane of the parietal cell, or any cell, GI cell for that matter, that faces the food side is your apical side. And this is anything that faces the blood side or the extracellular fluid is your basolateral membrane. So these are depicted here in the basal membrane, but this can also exist in the lateral membrane. So that's why it's basolateral. And so this is your parietal cell in another book. And they're usually depicted as a crocodile looking, which you can see here. So this is your basolateral side. This is the basal membrane. And so this one, the facing the food side, is your apical membrane. And there are three major factors that bind to the basolateral membrane that stimulate and regulate hydrochloric acid secretion. The first one is acetylcholine. We have acetylcholine binding to the M3 receptors. I described this to you. The second messenger is calcium. This can be blocked by atropine. I think you learned this earlier. Okay, so this is a classical inhibitor of ACH binding to its receptor. So this is a neurocrine signal. The paracrine signal is the endocrine signal is the next one here. This is the gastrin is from your G cells. ACH is from your vagus nerve. Gastrin is released from G cells, and it is recognized by a CCK receptor, CCK number 2 receptor, or CCK beta receptor. These two names are found in different books. So that CCK2 or CCK beta is a receptor specific for gastrin. And this binding here can be blocked by proglumide. And the second receptor is calcium. I mean, the second messenger is calcium. Now, the paracrine signaling is via somatostatin, via histamine, here binding to the H2 receptor, which can be blocked by cimetidine. Cimetidine is, I think, Pepsid AZ or Zantac or Tagamet, one of those over-the-counter drugs that are called, antacids, and they block the binding of, histamine or H2 blockers to their H2 receptors. So this can be blocked by cimetidine, and histamine comes from your ECL cells. The second messenger here is cyclic AMP. Then you also have factors that inhibit hydrochloric acid secretion. Paramount in these factors is somatostatin, which actually binds to the somatostatin receptor number 1 here. And somatostatin would inhibit the synthesis of cyclic AMP, the second messenger of H2 signaling here. Prostaglandins also inhibit acid secretion via inhibiting cyclic AMP. Epidermal growth factor binding to EGFR, prostaglandins binding to PGE2, somatostatin binding to this receptor, they all act to inhibit hydrochloric acid secretion by cyclic AMP. We will take a break about 3:30, okay, halfway around the lecture. Okay, so the inhibitor of the proton potassium pump by itself can be inhibited directly. And these are called proton potassium ATPase inhibitors. And the classical one is omeprazole. So these are also now found over the counter. So
Professor?
I showed you 3 secretagogues, right? First, I showed you acetylcholine, second messenger calcium. So if you only have a stomach with only acetylcholine as its signal, it will make acid by itself. Or if you have a stomach that has histamine binding to H2, only this factor will be sufficient to stimulate acid. If you have a stomach that only has gastrin receptors, it will again secrete acid. So each secretagogue by itself stimulates proton secretion. However, if you put them together, their simultaneous effects is much greater than the sum of their individual effects, which means that there is enormous interaction among these 3 factors. And these interactions are called potentiation. In other words, the presence of one alone would be sufficient to stimulate proton production, but the presence of 3, there will be feedback mechanisms so that they're really able to ramp up proton secretion by a lot if they're all 3 are together. And how does that happen? Okay, so this is how it happens. So this is a web of showing you neurocrine signaling, endocrine signaling, paracrine signaling. So let's begin with acetylcholine. So it stimulates the parietal cell, shown here. It stimulates the ECL cell, shown there, here, the G cell anyway, releasing gastrin, releasing peptide, and I'll talk about that later on G cells. So ACH stimulates acid secretion, gastrin secretion, histamine secretion, and inhibits the D cell. Now the D cell makes somatostatin. Recall that from the previous slide. So acetylcholine is very potent because it stimulates the positive regulators of acid production and it inhibits the negative regulator of acid production. Now let's look at the secretory product of G cell, gastrin. Gastrin stimulates G cells, ECL cells, sorry. G cell makes gastrin and gastrin stimulates ECL cells, shown there, and also stimulates the parietal cells, shown here. Now histamine is the main stimulator of acid production, as shown here by the numerous pluses here. So histamine, by its, after all of these, all of these interactions and potentiation, at the end of this, it's really the biggest stimulator of acid secretion. That's why H2 blockers alone are potent anti-ulcer drugs. These cells make somatostatin and it inhibits, shown here, parietal cells. It inhibits the G cell, shown here, and it inhibits the ECL cells. So the D cell making somatostatin not only inhibits the parietal cell, as I've shown you before, but also the G cell and the ECL cell. So I was just talking about the aqueous component. So in general, the factors that release hydrochloric acid from parietal cells also release pepsinogen from chief cells, so that their secretions are actually intertwined and interlinked. So when the chief cells receive acetylcholine, they will secrete the pepsinogen. If they receive acetylcholine or histamine, they will also release pepsinogen. So therefore, you time the release of pepsinogen so that it coincides with secretion of hydrochloric acid. So it increases the efficiency of protein digestion. The biggest stimulation for mucus secretion is also by these secretagogues, but the biggest contribution is by acetylcholine. And I'll show you also mechanoreceptors in the later part of this lecture. So I've just shown you that the gastric juice or the secretions of the parietal cell are so are voluminous, right? So, and there's a lot of protons there, such that the pH of the lumen shown here. So this is the food side, this is your blood side, as indicated here. So these are like your luminal region, and this is the region of the mucosa that's overlain with mucus. So you have actually your surface mucus cells and your mucous neck cells making copious amounts of mucus proteins. So what they do is they protect the gastric mucosa from its own secretions. They overlie that. It's like a gel. Okay, so an actively normal person who's actively eating don't digest its stomach wall, because a lot of it is due to the mucus, and of course to the regulatory mechanisms we just talked about. But if you look at the pH change, so the mucus creates a protective lubricating coat on top of these epithelial cells. And if you look at the gradient, if you have a microelectrode, going from the luminal side where there is actually a fair amount of flux and all the way close to the surface of the gastric pits, you find that microelectrode will tell you the pH is 1 to 2 in the lumen. But as you go closer and closer to the epithelial cells themselves, the pH actually goes higher. That is because the protons have a difficult time diffusing through the thick mucosal coating. In a way, you're protecting yourself from your own lethal secretion of acid. A pH of 1 or 2, you could put, probably a nickel there. Eventually, the iron, these metals will melt because it's so very acidic. Yet we survive that secretion because mainly because of this mucus layer. So when we examine this mucus layer, we found this gradient and the bicarbonate. So there is also copious secretions of bicarbonate. So as the parietal cells make a lot of protons, the mucous neck cells and surface mucous cells are actually also actively making bicarbonate that is secreted into the lumen side and gets trapped below the mucus layer. So that right above here, the pH is 7. And so the gastric cells here are comfortable. They're not fighting an acid of 2. They're just trying to, they have a pH of 7 here. So that they can tolerate. And you can see here, ladies and gentlemen, that really there isn't that much protecting you from an ulcer. This is your lumen with pH 1 or 2. You have mucus, you have one cell layer, and then you have your blood vessels below that. So that's why it's very easy to actually bleed out if you have an ulcer big enough that you lose a lot of blood through it. So C and D, so in this layer here, there's tight junctions. So the gentleman's question, so you have this apical membrane where the bicarbonate is being secreted. So this is the apical membrane here, and this is your basolateral membrane here. Okay, lateral, basal, lateral, lateral, basal, lateral. This is your interstitial fluid, and then this is a capillary that supplies the bicarbonate and takes away the protons from this immediate area here. So how do you get then ulcers? So under normal conditions depicted here, the damaging forces represented by enzymes and acidity is balanced by what I just showed you, mainly mucus secretion, bicarbonate, etc., here, prostaglandins, right? So because prostaglandins inhibit cyclic AMP, inhibit acid secretion via cyclic AMP. However, you can get ulcers because of H. pylori, which is a bacteria found in your stomach and irritates the mucosal lining. And this occurs when the damaging forces overwhelm protective defenses. So this happens, this is represented by increased damage and ulcer forms, or because of impaired defenses. So you get ulcers when you have infection, non-steroidal anti-inflammatory drugs, aspirin, cigarettes, all the good things in life, attacking your gastric mucosa here and resulting therefore in an ulcer. You could also get a significant number of people with ulcers is due to aspirin. And why aspirin? Well, this is your cyclooxygenase enzyme. Cyclooxygenase is required for formation and synthesis of prostaglandins and thromboxans. And aspirin, as depicted by this chemical reaction here, acetylates, one amino acid residue here in cyclooxygenase, and the acetylation is irreversible, and therefore these ones don't work. You don't have prostaglandins, you don't have thromboxans, or your reduced levels, and eventually you will get an ulcer. Okay, so let me introduce to you the pancreas before we take a break. Okay, so this is your pancreas. And you know your pancreas from all the famous people who have had pancreatic cancers. Unfortunately, it's incurable, and it's very, very important for endocrine function. You know that, for those of you who have diabetic relatives, the pancreas, it has a major endocrine function in making insulin and glucagon. Okay, so but we don't discuss this here. Dr. Gaspers will. Okay, this was towards the end. The pancreas has an exocrine function, and the main exocrine function is bicarbonate. The secretion is bicarbonate, and that's the aqueous secretion, and the organic secretion are mainly enzymes used for digestion of fats, proteins, and carbohydrates. So if you take and make an histological section of a pancreas, this is actually what you see. So it's like a salivary gland in a way. These are your acinar cells, and they make digestive enzymes. And these are your duct cells lining the pancreatic ducts, and they secrete bicarbonate, as depicted in a later slide. So most important component is bicarbonate, and then we have organic components. We have amylases to break up starch, a carbohydrate. We have a variety of lipases to break down fats, a large variety of proteases to digest proteins, and we even have ribonucleases to digest RNA and DNA from food. So we don't, we don't waste anything. If we eat, we partake of the carbohydrate component, the lipid component, the protein component, even the nucleic acids. Okay, we eat that and we absorb those. So this is just how, if you're an actively secreting pancreas, will work. So these are your acinar cells, and these are your duct cells lining here. And so as represented by these very colorful enzymes, they come from the acinar end. And the duct cells, what they do is they add sodium bicarbonate mainly to the mix, as depicted here. So the composition of pancreatic juice changes with flow rate, which is not a surprise to you anymore. So the x-axis, flow rate here, is the rate of pancreatic secretion. The y-axis, lo and behold, is ionic composition. And again, ionic composition of pancreatic secretion is a function of flow rate. At basal rates, you have this composition here. At basal rates, your pancreatic juice is composed mainly of sodium, chloride, and a little bit of bicarbonate. Bicarbonate greater, chloride less than plasma. You could confirm that, later. So what happens during when you're eating and the pancreas is stimulated? Well, it goes from sodium chloride secretion to sodium bicarbonate secretion, and with now an enormous volume. So this is the sodium and potassium are independent of secretion rate, and then the sum of the negative ions, chloride and bicarbonate, is always the sum of sodium and potassium. Because these ones don't change, their totals don't change, then the osmolality of the pancreas actually do not change with flow rate. So what is response? So we now know that bicarbonate is being secreted. So we have in the stomach, we had the parietal cell responsible for acid secretion. So here we have the pancreatic duct cell responsible for bicarbonate secretion. So this is the food side, this is the blood side, this is your basolateral membrane. So these are the major players again. So lo and behold, you have the same carbonic anhydrase reaction. And the big difference is that the chloride bicarbonate exchanger that was in the parietal cell found in the basolateral membrane is now located in the apical membrane of pancreatic duct cell. Okay, ladies and gentlemen, this is not the same genetic protein. It's not the same gene. It is the same family. So they do the same thing. They exchange chloride for bicarbonate. So here, remember, what is the problem of a parietal cell? If you're a parietal cell, what is your biggest byproduct? Bicarbonate, right? But you don't want to throw away your bicarbonate into the food side because you're ruining, your secretion. Has to go to the blood side. You use a chloride bicarbonate exchanger. Now, you're a pancreatic duct cell, what is your biggest headache? Your biggest headache are now protons, because your biggest secretion is bicarbonate. Okay, you want an alkaline secretion. And so this chloride, the bicarbonate then is exchanged with chloride. And then we have a recycling of your chloride. So we have a net secretion of bicarbonate. The protons are extruded to the blood side by the sodium hydrogen exchangers. Okay, because you want to put them away to the to the blood side. And so, ladies and gentlemen, this is CFTR. So CFTR is a kind of chloride channel that, as you know, in certain populations, you have cystic fibrosis, you have mutations of CFTR. So in people with severe phenotypes of cystic fibrosis, their pancreatic ducts are not working well because this is not working. So that's we throw away the protons to the blood side by way of the sodium hydrogen exchanger. So just as the parietal cell of the stomach had a very, very big problem, as a gradient, there is also a gradient against bicarbonate secretion into the lumen, the food side. However, there's a downhill gradient for chloride, so this one operates by this chloride downhill gradient. So this downhill gradient powers the uphill gradient of bicarbonate. And to maintain electron neutrality, we have sodium and potassium ions passing through between cells. Okay, so that's how sodium and potassium go from blood side to food side, and then water actually would follow in this fluid. And water goes through via water channels, which I'll be describing to you how this happens in the absorption side of our lecture next week. So water then would contribute to the enormous amount of fluid that's being secreted by the pancreatic ducts. The hormone that controls the secretion, okay, so this is a human volunteer. This is time, and this is the concentration of electrolytes in the pancreatic secretion. So this is the normal levels of amylase, potassium, sodium, and so that's, apologies. So the moment secretin is injected into this volunteer, you have 100-fold increase in volume of pancreatic secretion. So secretin is a very potent releaser of bicarbonate secretion. Not only do you have an increase in volume, you also have an increased concentration of bicarbonate. Secretin, as you will find out in later slides, actually is released by chemoreceptors in the duodenal lumen, which detects extreme acidity coming in from the stomach. And so this duodenal cell senses that, and I'll show you this in a later slide, and then would make secretin so to neutralize the acid that's coming in from the stomach. Amylase is released by a different hormone, CCK. And in conjunction with that, acetylcholine, as depicted here. So this is the food side, this is your blood side. So these are your enzymes, and these are made in the pancreatic acinar cells. And they are actually released mainly by calcium signaling when CCK is present and acetylcholine is present. So acetylcholine binding to the M3 receptor would release calcium, a second messenger, phosphorylate these regulatory proteins. There's a fusion of these granules containing enzymes with the apical membrane. And then once these vesicles join the apical membrane, they empty their contents into the lumen, which is then washed out by the secretion from the pancreatic ducts. So the main regulator for enzyme secretion is mainly acetylcholine, as depicted here. And this one, CCK, binds to the CCK1 receptor in the basolateral membrane of this acinar cell. So the CCK, remember, is a cousin of gastrin. So CCK binds to CCK1 or CCK alpha. I will have this in later slides. Gastrin binds to CCK2 or CCK beta. I will explain why CCK is a minor controller of enzyme secretion, and its receptor is CCKA, and their second messenger is calcium. VIP and secretin actually potentiate this CCK effect. And then overall here, somatostatin, as depicted in my drawing, inhibits adenylyl cyclase and affects and slows down secretion of enzymes by acinar cells, as depicted here in this graph. And CCK, where do CCK and secretin come from? They're actually made in duodenal cells, and I'll show you where, which part in a later slide. They're made by secretin by S cells, CCK by I cells, and these are found in the duodenal mucosa, and secretin and CCK are then secreted from those mucosal cells into the blood side. Okay, so let's take a short, a 5-minute break so you can stretch and I can stretch, and I will pause this, and I hope I don't ruin anything. There you go. And if you have questions, you could, well, one of the things I will do actually is if you have questions, you could ask them to me, or I will actually summarize them, put them all together and cover them in the review on Friday. It's probably better because some of you ask the same types of questions anyway. So I will integrate them. You could email me the questions you'd like me to cover in the review. And for those of you who are, online, you probably heard me as well. So you could email me your questions, and in particular, when you email me questions, attach the slides, embed the slides that puzzled you. That way, I know exactly where to go to. Okay, go ahead. Okay, yeah, thank you. Thank you for for attending the lecture. I mean, one of the things that I'm scared of as as a lecturer, as a professor, is especially in these COVID times, I'll be lecturing to an empty hall. It's really not good. So thanks. Okay, so let me restart. You guys can hear me all right? Right, so, okay. So it defaulted to the very beginning. Okay. Okay, so can the online students also hear me? I don't see or hear any chat problems. Okay, so let's begin. And so here in this last slide, this was your acinar cell. Remember in that GIF depiction, the acinar cells was making the enzymes, the duct cells was making the bicarbonate. And so that's the same story. We will have that sort of theme is also the same as in the salivary gland, where the acinar cells make enzymes and the duct cells are making the aqueous secretions. So from the pancreas, we now go to the liver. The liver, excuse me, actually does not make hormones or enzymes, yet it plays a critical role in in GI physiology. And the major reason for that is alluded to in the second slide, because of the difficulty that lipid nutrients, the lipid component of our food, presents as a substrate of digestion and absorption. In fact, that there's probably I would say 5 to 8 times more slides dedicated to lipid digestion and absorption than there is for carbohydrate digestion and absorption. And there's ample reasons for that. And so here we begin with the liver. So these are your hepatocytes, and they secrete bile. And this is represented by this green fluid. And then these are essentially bile ductules, and they collect into bigger and bigger ducts, eventually into the main bile duct that convey the secretions of these numerous hepatocytes, eventually into the bile duct and the gallbladder that's shown in the next slide. So bile is made in hepatocytes and then is collected in the gallbladder via these bile ductules. There are also other functions which I will be covering today, mainly the metabolism of bilirubin, which is a breakdown product, a pigment of your hemoglobin, and the storage of lipid and soluble vitamins, which I will cover next week. So from the liver, between meals, when you're listening again to my lectures, the sphincter here, sphincter of Oddi or Oddi, is closed. So your liver is happily making bile, and that bile is stored in your gallbladder until such time when we need it. So bile right now is in the gallbladder, but is released during meals, which we will be discussing. And bile has aqueous and organic components. It's nothing new anymore to you. The aqueous component is relatively simple. Its main, is bicarbonate. So all of these secretions is bathed in a bicarbonate-rich fluid. And so bile is slightly alkaline in nature. And it's also partly regulated by secretin, so that it increases the volume of these secretions when secretin is around. However, the organic component is really the most important component. And there's 5 components. First is bile acids. Second is cholesterol. Third is phospholipids, about 20%, bile pigments, 1%, proteins, 5%. So let's focus on bile acids. Some of your books will call them bile salts. They're actually the same, because in these acids they form salts with some cations. So, okay, so secreted bile acid is a polar or amphipathic molecule. So this is your bile acid. The gray part is the hydrophobic phase of the bile acid. The hydrophilic one are these are presented by these hydroxyl and carboxyl groups. And that's the hydrophilic phase. So it's an amphipathic molecule. It's, so it's polar. It has one side highly charged, the other relatively hydrophobic. And so this bile acid is actually made up, I don't have time to show you how they're made, but they're made up from cholesterol, to which hydroxyl groups and amino acid groups are added. And the cholesterol is their backbone. And so hydrophilic groups of bile acids sequester among themselves and associate with each other or other lipids. So you could actually see this, so you can imagine this to be an aqueous medium. And so your lipids are actually sequestered within structures bordered by bile acids. Because by biophysical rules and entropy rules, it's really only the hydrophilic parts of the bile acids that can interact with water, whereas the hydrophobic part actually forms the part that dissolves these lipid products, as depicted here. It is the purpose of this amphipathic type of molecule. So what are bile acids for? Well, they're mainly for digestion of lipids. And it's not that they really participate in the actual breakdown, but what they do is, so ladies and gentlemen, when you actually go and celebrate your wisdom now in GI physiology and eat ice cream in the cafeteria, the cream part of your ice cream will be in your stomach and in your luminal fluids of your upper intestine as ugly looking large fat globules. And when your pancreas releases enzymes to try to digest them, because they are aqueous and hydrophilic, they are only able to nibble at the surface of a large, ugly looking fat globule. So the function of a bile salt is to emulsify this fat globule. It's like a detergent. And it breaks them down into emulsions, emulsion droplets, very, very, very small. Therefore, what they're doing is they're just expanding the surface to volume ratio of these lipids, so that now your enzymes can work more efficiently and are able to digest them, your lipids more efficiently. Their other job is absorption. via micelle formation, which I will be describing in the lipid absorption section. Okay, so for now, we're going to talk about the digestion component, and absorption. I will allude to this until we see absorption in the later part, I mean next week. So the second organic component of bile is cholesterol. And the cholesterol that our body makes is secreted by hepatocytes, and bile is its only route of excretion. So this is your, this is an example of a micelle, and these are cholesterols that are found, in these micelles. And they actually, essentially, these bile acids solubilize, again, cholesterol and all of these lipid products, as depicted here. Now you have phospholipids. 20% of bile is phospholipids, and and lecithin is the most common type. And they have these are like phospholipids. So this head group here is your hydrophilic component. These two lines here represent your hydrophobic component. So if you look at it here, then the polar components are exposed to the aqueous component of the luminal solution, right? So if you can imagine this one as a ball, as a droplet, an emulsion, a sphere, the ones that are exposed, the surface, are actually hydrophilic molecules. Then we have bile pigments, which are the degradation products of hemoglobin. This is mainly bilirubin. And what hepatocytes do is to remove bilirubin from the blood, conjugate it, and secrete it into bile. We, of course, don't have time to discuss this in detail. That's why if you have issues with your liver, you get jaundice, which is mainly your skin turning yellow. And why? So how this is happening? So these are your red blood cells now being destroyed because they are being replaced. And so the hemoglobin is broken down, bilirubin is released into the bloodstream. And then in the bloodstream, this bilirubin is bound to albumin. And they go through your systemic circulation. And bilirubin is taken up from the blood into the hepatocytes. And inside the hepatocytes, they're linked to glucuronic acid by this enzyme called UDP glucuronyl transferase, or for short UDP-GT. And then it's secreted along with bile. And once it's in the lumen, okay, so this is now secreted, and so this represents your small intestine. This inside here is your food side, outside here is your blood side. So the bacteria, the microbiota that I alluded to in the small intestine, convert this conjugated bilirubin into more hydrophilic urobilinogen. Otherwise, it would just basically diffuse through the mucosa and go back again. Then it becomes more hydrophilic, therefore, and even becoming more hydrophilic with stercobilin, and then you defecate this in the feces. And that's why this actually makes the color of your feces, if you're normal, to be colored, brown. So this is how you get rid of your bilirubin. So let's go back to bile. So we know that bile is very important for lipid digestion, as well as absorption. So it's prevented from entering the lumen where it can do its work by this sphincter of Oddi. So between meals, like when you're listening to GI lectures, then the sphincter of Oddi is constricted and your gallbladder is relaxed. So bile from your liver would then go by default into the gallbladder. And so between meals, you have this vasoactive nerves, which has VIP, or vasoactive intestinal peptides. They are between meals, they actually inhibit gallbladder contractions between meals. And the sphincter is actually closed by default. So this is your lumen, this is your duodenum, this is your stomach is here, and this is your intestine. So this is a more proximal part of your intestine. And this is where the gallbladder connects and empties into the lumen of the intestine. So the gallbladder is relaxed between meals because the VIP is there. And think of VIP as a relaxing neurocrine secretion. So what happens during a meal? So during a meal, you celebrate your wisdom in GI physiology. You eat your favorite steak. And so then it's releasing all these nutrients. The nutrients are detected by your eye cells, which make CCK. CCK then is released from the eye cells into the blood side. Some CCK would travel via the blood as an endocrine signal, bind to CCK1 receptors in the wall of the gallbladder, and initiate smooth muscle contraction by itself as an endocrine signal. ACH is also detected by vagal afferent nerves, whose nerve endings have CCK1 receptors. These CCK1 receptors detect the presence of CCK, which tells them, hey, there is a lot of food in the lumen of the duodenum that needs to be digested. So the CCK would send signals to the brain, the central nervous system, and vagal afferents, the ACH here would be a neurocrine signal that would also stimulate the gallbladder to contract. This vagal afferent would send ACH to the wall of the gallbladder to make it contract, but would send VIP to the sphincter to make this sphincter relax. So contract there, relax here. And this no VIP, that means actually nitric oxide. It does not mean no VIP. It means nitric oxide and VIP. So here, therefore, dietary fat via CCK and ACH tells the gallbladder to contract in order to release bile for fat digestion and absorption. So now, this is your bile in the duodenum, in the jejunum, and ileum. And in the more proximal regions, I mentioned to you, they emulsify and digest fats. They are also absorbed, they also participate in micelle formation and therefore in fat absorption in the jejunum. So digestion and absorption of fats occur here. Now your bile is now here. Do you want to defecate your bile or do you want to recycle the bile? The gallbladder concentrates bile, but the gallbladder is actually too small to emulsify all the fats in a single meal, especially if you eat a lot of ice cream, because you learn a lot of GI physiology in a single day. So you reward yourself, and so now your bile is then recycled. And depending on the amount of lipids that is found in your food, it would be recycled several times so that you can use it. Less than 5% of bile is actually reaches the colon. So this process, ladies and gentlemen, is called enterohepatic circulation. This is how important your bile is for lipid digestion. And here, once the gallbladder is only acting here as a storage organ. Once it has released its contents of bile and bile acids, it doesn't participate anymore in enterohepatic circulation. So now we have a stomach that's secreting acid. We have a pancreas that's secreting bicarbonate. We have liver that's secreting bile acids, even bicarbonate. And so if these were organ systems that were operating independent of each other, you will not be able to digest and absorb your food properly. They need to be coordinated. So you need to coordinate your secretory processes. And it does involve hormones and the central nervous system. And when it involves nerves, there are two kinds of neural control. If it involves the central nervous system, as shown here, your books call it a long reflex or a vagovagal reflex. If it doesn't involve the central nervous system and the signaling is only among nerves within the GI tract, the enteric nervous system, then that is called a short or local reflex. So again, these are two terms used by different books and different websites. So those are the categories by which, so there's in a way three types of control: hormones and long reflex or short reflex. Now there are also three phases in the control of secretion. The first phase is called the cephalic phase because it's named after the site of receptors initiating the secretory process. Okay, so this is when you're thinking of good food. Okay, if you're driving home and you know that your partner is cooking your favorite food because you just learned GI physiology, then you will be thinking about that alone would already initiate the process of secretion. So this is when you seriously think of food or when your senses are stimulated by food. Gastric phase occurs when actually food enters the stomach, but not yet the intestine. So, remember that food will be processed in your stomach for a while. So food enters the stomach, it has its own chemo and mechanoreceptors there. You can actually cut the vagus nerve and you will be normal. Okay, you could actually digest because of these short reflexes and hormones being initiated by the gastric phase. And then the intestinal phase, as the name implies, when the contents of the stomach is emptied into the duodenum, activating its chemo and mechanoreceptors there. So let's talk about the first gland that we talked about today, the salivary gland. And then this is the graph in a different format. Remember the smell of food, thinking of food, would stimulate the parasympathetic nervous system. Remember that sleep, you should not salivate when you sleep. Otherwise, it's a grounds, as I said, for breaking up. And so, because this should inhibit salivation. Okay, so stimulation of this, if you smell good food, if you think of good food, even if you taste good food, if you chew actually cardboard, ladies and gentlemen, you actually will salivate. You don't have to do that experiment. All you need to do is believe me. And then for this, we talked about the transmitter being acetylcholine, mediated by PNS, blocked by atropine. And so because it involves the central nervous system, remember, cranial nerve number 9, the medulla, and salivatory nuclei in the medulla. So these are long reflexes. So this baby, when given that food, actually is using long reflexes to react to the food that he or she was given. Probably not very well tasting. Okay, so what about gastric phase during the cephalic phase? Gastric secretion during the cephalic phase. Okay, so cephalic phase, again, when nothing is, you're just thinking about food. So that alone you're thinking of food or smelling or the act of chewing would stimulate the vagus nerve and release acetylcholine to the interneuron that's connected to the parietal cell. The parietal cell would then, and from the interneuron to the basolateral membrane of the parietal cell, it would release ACH, and then the parietal cell would release protons through the apical membrane. So you have parietal cells. I did not depict here Chief cells. I did not depict mucous cells. But this same pathway is also regulating those cell types. What about G cells? Well, the same is happening. So you have acetylcholine released on interneurons. And then the interneurons that are innervating G cells in all mammals use gastrin-releasing peptide, not ACH. Okay, so this is GRP that is released on the basolateral membrane of G cells by specialized neurons bringing signals from the interneuron to the G cell. When we think of food and when we're going home and driving home, we absently, mindedly think, oh, okay, there's really nothing going on in my GI tract. But people who have studied this, in behavioral and neuroscientists who have studied this, know that the cephalic phase already accounts for 1/3 of the stomach secretions. So basically, when you reach home to partake of any food that is for you at home, you're ready. Your stomach is already pH 1. Okay, you're ready to jump in and enjoy the steak to celebrate, your learning of GI physiology. So, what about pancreatic secretion and the cephalic phase? So, if you remember, there's nothing entering the stomach. You only are thinking. Okay, you're thinking and smelling maybe. So, what is released here? What is the only hormone that's released? Gastrin, right? That's the only hormone that's released. So gastrin, there's no secretin, so nothing. And I remember that I mentioned to you that acetylcholine can also go to the acinar cells and actually stimulate the acinar cells into secreting small enzymes. Well, without any secretin, there's no increase in volume. So there is actually a release of enzymes, very low volume and very high concentration of enzymes, during the cephalic phase. Now, the only hormone that's released is gastrin. Now, gastrin, as I mentioned, is a cousin of CCK. So gastrin is floating around and binds to the acinar cells. The acinar cells are specialized for CCK. Gastrin also goes to the gallbladder, but the gallbladder has CCK1 receptors. The acinar cells here have CCK1 receptors. The receptor for gastrin is CCK2, as I mentioned in the parietal cell. So gastrin binds to parietal cells to the CCK2 receptors at high affinity. But because it's a cousin of CCK, it can bind at low affinity to the CCK1 or CCK alpha receptors and confuses them a little bit. And so therefore, you actually have some secretion of acinar cells, even if their main signal, CCK, is not yet released. And remember those vagal afferents that the CCK receptors bind to, that are activated by CCK? Well, those CCK1 receptors also respond to gastrin, but weakly. So there is again a weak signal bringing ACH here. So all in all, there is really just low binding, and there is like a false signal at this stage. There's no CCK being released, only gastrin masquerading as CCK. Okay, so now we are in the gastric phase. So the salivary secretion is still mainly the long pathway. So now food is in your stomach and would stimulate mechano and stretch receptors there. So these are depicting stretch receptors. So let's follow this one. So when food enters the stomach, the chemoreceptors fire, the stretch receptors fire. And there's this local network, this local reflex, interconnects this intrinsic plexus to the interneuron, releases ACH there. Okay, so it releases ACH to the interneuron. And this local reflex releases ACH on the G cell. So you also see this stretch receptors sending ACH here and releasing ACH on the parietal cell. So this then is your local reflex or your short reflex. There is no, at this stage, you can cut the vagus nerve and you can still happily have relatively normal function. However, some stretch receptors actually connect with vagal afferents. So those signals go up to the central nervous system, and then they come down and release acetylcholine here, releasing acetylcholine there. And then this long reflex releases acetylcholine in the interneuron that is innervating your G cell. And the long reflex releases GRP on G cells in the stomach. Okay, so you need to understand the conditions by which GRP is then released. So here, just a long reflex releases ACH on the interneurons, but then GRP on G cells, and the long reflex releases ACH on the parietal cells and on the other cell types. So I alluded to the fact that there's also chemoreceptors. So you have G cells responding via their chemoreceptors on their apical surface and would release gastrin. Your parietal cells have been known to respond to dietary calcium, to excessive amounts of alcohol and caffeine. That's why if you have a problem with ulcers or hyperacidity, your gastroenterologist would tell you to minimize, in fact, avoid intake of alcohol and caffeine. So what about the pancreatic secretion in the gastric phase? So in the gastric phase, what hormone is released? Only gastrin, right? So there is nothing else. So the release of gastrin then is just the same as what happens during the cephalic phase. You have low affinity stimulation by hormonal means of the CCK1 receptors in the acinar cell of the pancreas, and then low affinity binding to CCK1 receptors in this vagal afferents, which go to the central nervous system and bringing down a small amount of ACH released on the acinar cells. So then you have enzyme release, but no secretin release. So now we are in the intestinal phase. And so salivary secretion is still regulated by the autonomic nervous system. So throughout, the salivary secretion is regulated by the vagovagal reflex or the long reflex. And if I ask this question in any exam, some people still do not answer me correctly. And I don't understand why. Okay, so gastric secretion in the intestinal phase. So intestinal phase, as the name implies, food is already in your small intestine. So gastric secretion is still stimulated when the pH in the stomach is greater than 2 to 3. Food buffers the stomach. Okay, so when you eat food, if you have ulcers, sometimes your gastroenterologist would tell you to eat food frequently, because food would actually buffer the acidity there. So in the early part of your digestive process, the pH, because of the presence of food, the more you eat, the higher the pH actually in your gastric lumen. It goes from pH 1 to 2, then food arrives, and then it increases. And for so long as you're enjoying your meal and eating, food stays, pH remains high. And then towards the end of a meal, you're already watching March Madness there. Okay, so then the pH in your stomach lumen will drop because there's no more food to buffer the gastric secretions. Then acid production would be inhibited during when the pH is actually low. So during the intestinal phase, we have when the duodenum is being distended, there are mechanoreceptors in the duodenum, and there are G cells in the duodenum, and they release gastrin from the G cells in the duodenum to influence acid secretion in the parietal cell. They also release a hormone called enterooxytocin, which is a gut hormone that enhances acid secretion by the parietal cells. So this is another hormone that is released only during the intestinal phase. There's also chemoreceptors in the lumen, facing the lumen of the duodenal mucosa. So they release, they sense digested proteins in the food side, and then they would release enteroxytocin into the blood side. The G cells in the duodenum would sense nutrients in the lumen side and would release gastrin to the blood side. So this is still stimulation. So now you are done with your dinner. You are enjoying, men's and women's NCAA playoffs. And so you have inhibitory effects. So there's three factors that mediate this. Now there's less food to buffer your stomach, so the chyme that's entering the duodenum becomes highly acidic. So now this is your stomach contents entering the duodenal lumen. So now you have lipid products and acid secretions. The acid secretion stimulates chemoreceptors in your S cells. The S cells make secretin. They go tell the duct cells to make and release tons of bicarbonate in copious volumes to neutralize the acid entering from the stomach. Secretin actually also inhibits gastrin release by G cells. So there is an arrow here that you are free to make going into, and I didn't because it would just be too confusing, getting and inhibiting the G cells from releasing gastrin. So secretin stops acid twice by releasing a natural antacid bicarbonate and by inhibiting the release of a stimulator of acid secretion. And then when there's less food in the duodenum, then the mechanoreceptors are not firing, and it would inhibit or reduce the release of enteroxytocin, which, as you know, stimulates acid release. So the inhibitory effects, okay, so now there's no food remaining in the stomach. You have D cells here. Okay, so this is your D cell. It's called SS cells by some books, or somatostatin cell by some books. And so this is, ladies and gentlemen, your food side. This is your blood side. So the increased acidity would stimulate the D cell into making somatostatin released onto the blood side, and the blood side would, by paracrine signaling, would go to nearby G cells and inhibit them from making gastrin. Also, the increased acidity would stimulate the D cells, depicted here as trapezoids, releasing somatostatin and inhibiting parietal cells here, shown by this hexagon, from releasing more acid. And I just brought this in because this is actually a fairly important slide. Wherever you go, if you're entering into dental physiology, you will see me again, and you will see this again. If you go into medical school, you will see me again, and you will see this again. It's going to be boring by the time. No, because I'm going to ask very difficult questions over there. Okay, so your acetylcholines do the same thing. you stimulate. So you basically study this web, and you actually add more arrows to the web that I shown you. So now based on the previous slide, we know that increased acidity in the lumen would stimulate the D cell, and the D cell would release somatostatin, which inhibits the G cell. It also inhibits the parietal cell quite potently and inhibits the ECL cells. Okay, so you have all of these webs that you can create in addition to my own arrows here. So what about the pancreas in the intestinal phase? So this is your stomach, this is your pancreas, this is your duodenal lumen, this is your sphincter of Oddi. So the presence of acid in the duodenal lumen releases secretin. We just talked about that. The presence of fats would stimulate I cells, releasing CCK, and CCK would stimulate secretion of enzymes via the acinar cells and via ACH. Here, vagal stimulation. Secretin causes release of bicarbonate secretions. So the bulk of human pancreatic response actually occurs in the intestinal phase. So the reason the pancreas responds during the cephalic phase and in the gastric phase, it's because gastrin is malingering as a CCK. And so therefore, some of the receptors by low affinity binding gets excited, but not too much. So you have 20% release at that time. So then, as I alluded to, you have this aqueous response where acid induces release of secretin from S cells in the duodenum. And then the organic phase, dietary fats, amino acids, stimulating I cells to release. And then you have this CCK via afferent nerves of the vagus causing ACH release, and CCK as a hormone binding to its true receptors in the acinar cells. So over here then, this actually is, we're done with the secretion phase, and we are transitioning into the intestinal secretions and some introduction of the small intestinal function. Okay, so as I mentioned, you can email me questions, and I will put together those questions in the review. If you have, it would be better if your questions are accompanied by specific slides, because then we have many slides, and it's better if you tell me, like for example, we mentioned CCK all over the place. So what part of the CCK don't you understand, or would you like more clarity? And then you send me that slide, or more than one slide, but not too many. Okay, so we're transitioning into digestion and absorption, and you've seen this before. So we have 1 to 2 liters of saliva, all of these secretions. We have 9 to 10 liters into the small intestine, and then the small intestine absorbs about 80% of that, the large intestine about 20% of that, so that very little under normal conditions go into the stool. Most nutrients are absorbed in the duodenum and jejunum. Most water and ions are absorbed in the jejunum. The ileum, the most distal part, where the bile acids are absorbed, they absorb bile acids during enterohepatic circulation, water, ions, and again, vitamin B12. The colon does not absorb nutrients. It's specialized in absorbing water, the remaining water and ions. So the task of all of those secretory organs is to digest food to absorbable units. Now, the task of the GI system, the second half of the GI system, is to absorb hydrophilic nutrients across membranes. Okay, so what is the biophysical problem of a hydrophilic nutrient in being absorbed? The biophysical problem is it doesn't diffuse through the lipid bilayer of your intestinal cells. Yes, it is very easy to digest, as I alluded to, your apple, your rice, your potatoes. Okay, it's very easy to digest your steak. But the products of, they're now floating in the aqueous medium. And now they're releasing nutrients. Because they are hydrophilic, then their products are they're also mostly hydrophilic. So there's a problem. Whenever you have, you will learn this also in renal physiology, is if you have a hydrophilic molecule, then the apical membrane is not ready to absorb that. So it requires numerous transport systems. So the biggest problem of hydrophilic nutrients is not digestion. It's during absorption because it requires transporters. Okay, so this is your sushi. Okay, so to celebrate your newfound knowledge in GI physiology, you stop by your favorite sushi store to supplement the steak, and then you eat your sushi. And the rice has a lot of starch, and so amylose is straight starch. Amylopectin is branched starch. So your pancreatic amylase is busy cleaving them into small saccharide molecules. Maltose, maltotriose, glucose oligomers. Okay, this alpha limit dextrin is a saccharide that is branched. Then in the apical membrane, you have your brush border membrane enzymes. So you have maltose and maltotriose. They are digested to liberate free glucose molecules. Your alpha limit dextrin is a little bit difficult to digest. The glucoamylase would digest away some of the glucose molecules, and isomaltase would take off this branch chain to liberate this trisaccharide and, more glucose molecules. And then these then basically chop this up more to liberate glucose molecules. So from your sushi, most of it just becomes free glucose molecules. Very simple. It's all occurring in the most of this activity is occurring in the lumen. This is occurring in the lumen, but on top of the mucosa. Okay, it's still occurring. So now this is your, ladies and gentlemen, this is your lumen. This is your lipid bilayer, and this is your cytosol. This is your sucrase, this is table sugar, and your isomaltase. So you have your table sugar being presented there because you have ice cream, right, to celebrate your newfound knowledge in GI physiology. You have a ton of ice cream, a ton of sucrose that's digested to liberate glucose. Oh, no, sorry. Glucose here and fructose. Glucose is absorbed by a sodium-dependent process by this transporter called SGLT1, sodium-dependent glucose transporter 1. This is active and it's energy dependent. Fructose is absorbed by a facilitative transporter. In other words, if there's a lot of fructose inside, it will go back outside. This is facilitative. Okay, it depends on concentration gradients. Glucose never goes outside because the sodium gradient is reversed. It can't go outside. And so you can concentrate your glucose in the cytosol against a concentration gradient via SGLT1, but not against via GLUT5. So fructose goes in via GLUT5 or glucose transporter 5. There are maybe 2 or 3 SGLTs. There is one major one in the kidney, and it will play an important part in your lectures in the kidney. There's many GLUTs, GLUTs all over your body. GLUT5 is just a glucose transporter that evolved in evolution, okay, to recognize by high affinity fructose. Okay, so now glucose and fructose are absorbed, and this is your table sugar. What about lactose? Is milk sugar. Here enjoyed by your famous, you probably not recognize this group anymore. This was my children's generation, NSYNC, I think. So lactose is digested by lactase and liberates galactose and glucose. And then galactose and glucose compete for SGLT1's attention, so they can be absorbed from food side, lumen side, to the cytosol. These are very highly selective. The affinity of SGLT1 for fructose, very, very low. The affinity of GLUT5 for glucose and galactose is also very, very low. I mean, they can be absorbed, okay, but it has to be in pharmacological quantities here. So this is depicted in here as so this is your intestinal cell, food side, blood side. And I already described these two processes. So glucose and fructose, once they're in the cytosol, they exit the cytosol from cytosol to the blood side via GLUT2. GLUT2 is glucose transporter 2. As I mentioned, there's 14 to 15 GLUTs all over your body. So GLUT5 is the apical fructose transporter. GLUT2 is your basolateral glucose fructose transporter. Okay, so lactase. So when you go to the grocery store now, you look at milk, and there is an expensive type of milk that's called lactate. So what is so special about this expensive kind of milk? Well, lactase is present in children of all human ethnic groups. You have lactase deficiency, or LD, congenital lactase deficiency, is extremely rare. But lactase is often lost in adults of certain ethnic groups. So, for example, here it depicts lactase deficiency in Western Europe and in the United States of America, that is populated by people mostly from Western Europe. This group, ethnic group, evolved using milk as part of their daily nutrition. So they evolved so that their adults still contain lactase. So if you are of Western European origin, or even most of Europe, then in all likelihood, you are actually equipped to digest lactose. That's also true for Australia, because Australia is populated by people from Great Britain. And they all share this pastoral history where, they get their food from mammals they put out to pasture. There's Now, these are ethnic groups with very high incidence of lactase deficiency. So that's why I need to buy the more expensive type of milk if I ever, because I belong to this group, unfortunately. And if I drink this, and I did one day, and I had to run around all over the place looking for a place to socially do something. Okay, so however, because milk is now more and more present in our diet, there is lactase persistence increasing in all populations. Now, there is a group in Africa, the Maasai group, that also uniquely shares a pastoral history. They actually is a group, a subgroup of African populations that can still digest lactose because of the fact that they raise in their evolution, their cultural evolution, they relied on cows for milk. And this is a rough distribution of this deficiency. Now, in our last topic here, we're going to talk about the protein digestion in the small intestine. Okay, so we now are into proteins. We don't have to have proteins in our diet. Oh yes, go ahead. Mm-hmm. Okay, sorry. Congenital lactase deficiency is a mutated gene. However, the adult kind of lactase is not. It's a regulatory kind of deficiency. That's why that's why there's compensation, and this persistence is now evolving actually all over the world, because Western culture essentially is now spreading all over, and there's McDonald's all over China, and the number one deficiency is Oriental groups, but the fact is more and more mainland Chinese children are actually drinking milk, and so their lactase would be present. So it's a factor of inducibility. Yeah, if I actually gradually I can ramp myself up probably to have lactase eventually, but I don't want to try it. So okay, so luminal protein. I will try to find out more about that because that's an interesting. Yeah. Okay. So one of the things. Okay. So we get our protein from the steak that you're going to eat. Okay, because you now know a lot of GI physiology. You will be celebrating together with sushi. If you were to not actually have any protein, you'll still you can barely survive. Now, we actually require, we don't have to have any carbohydrate at all. Not at all. Okay, if you go on a diet, and we're not, people ask me about diets all the time. But what I'm saying is you can live without carbohydrates, but you cannot live without proteins, because your enzymes that you are secreting is made of proteins. Okay, the cells that line your intestines, your stomach, they're actually being replaced all of the time. You can just imagine almost all cells, that line the GI tract, they have a lifetime. The reason for that is they're exposed to really challenging environments. And you imagine if you're based in half of your life in very high acidity, you have enzymes that, trying to digest you. So eventually the body has evolved to replace this. So we lose about 50 grams of protein per day from these secretions or slab cells or other losses. So, if you have situations where you have famine, you need to send protein. That's the basic part. Because if you don't have protein, they will eventually die, especially children with a high turnover rate. So if you're sending aid, for example, to Ukrainian refugees, nutritionally, you got to send protein. You don't have to send them sushi, but you do need to send them chicken wings. Okay, so, okay. So protein hydrolysis begins in the stomach by pepsin active in the low pH. It's inactive in the duodenum, and we talked about this. But the bulk of your dietary protein, your steak, is actually digested by pancreatic enzymes. So how does this work? So your pancreatic enzyme releases these plethora of proteases. Trypsinogen, chymotrypsinogen, proelastase, you don't have to know, you need to know what they are, you don't have to know what they digest. So these are precursors of enzymes. The pancreas, as a way of self-preservation, do not activate these enzymes. So one of the most lethal diseases that you can have is if you have somebody punches you in the pancreas and your somehow on your abdomen and your pancreas gets injured, and there is some activation of these enzymes in your pancreas gets digested. So you never want this out. So we have evolved a way to do this. The mammals have. So in your intestinal cells, the lining of your epithelial cells, you have enterokinase. It's like, and its main task is to activate trypsinogen to release trypsin. And then after that, you have autocatalytic activation of that, and then trypsin then becomes activated. Enterokinase is resistant to digestion. Okay, so after you use your trypsinogen, other trypsins can digest trypsinogen. Okay, they can digest each other. These are proteins. So you keep on pumping more pancreatic enzymes, but enterokinase stays there. It stays there to keep this enzymatic digestion, because this is where your proteins are being digested. So now this is the steak that you're gonna have tonight. You're gonna tell your partner or your parents how much GI physiology you now know. You explain salivary secretion, pancreatic secretion over dinner, and they'd be so delighted to hear about this. And then, so these are your enzymes. They release oligopeptides and then di and tripeptides. Okay, so this is now shorter peptides. And then you have, oligopeptidases in the apical membrane. You have, just as you had, oligosaccharidases lining the apical membrane, you also have oligopeptidases that digest long chain peptides into shorter chain peptides. And then you have also brush border that peptidases that digest away di and tripeptides into free amino acids. It's just like your carbohydrates found in the apical membrane. And now you have amino acid transport proteins. So you have actually about 8 amino acid transport proteins found, and deficiency in them, you can lead to some form of if you have a mutation in some amino acid transport proteins, you actually have shortages of amino acids. And I forgot to mention that if you have a mutation in your SGLT1, you can barely survive. In the olden days, that was a death sentence. Now there are children that don't have SGLT1 who are actually surviving. But can you imagine walking around not being able to eat glucose or anything glucose? I mean, glucose is all over the place. We breathe and there's glucose. So, it's a difficult life, but now they're able to do that, mainly because they eat primarily fructose and taking advantage of GLUT5. So we have also a large number of amino acid proteins, transport proteins, because amino acids are hydrophilic. They need transporters to go from an aqueous medium to an aqueous medium while crossing a hydrophobic barrier. Lo and behold, we also have a peptide transporter. It's a di and tripeptide transporter. So you can so these are specific for certain families of amino acid transporters. So for example, ladies and gentlemen, you can have a cationic amino acid transporter. You can have an amino acid transporter that is specialized for anionic amino acids. Some specialized for neutral amino acids, etc., etc. So, but there's only one peptide transporter, and it is promiscuous. Can you imagine the types of combinations of di and tripeptides there are? This peptide transporter will recognize it and absorb it. And that's depicted here in this last slide that I will have for the day. Food side, blood side, cytosol. So this is only unique for proteins. There is no di and trisaccharide transport system for carbohydrate products. Okay, so so basically then, this di and tripeptide is recognized by this protein that's called PEPT1. Peptide transporter 1, because there's another peptide transporter in other organ systems. And it's powered a lot of these amino acid transport proteins, my apologies, are powered by a sodium gradient, just like SGLT1. They're powered, they're bringing amino acids from low to high cytosolic concentrations, and it's powered by a downhill sodium gradient. So the bulk of them are sodium dependent. This, ladies and gentlemen, is proton dependent. And the proton dependency is generated also by a sodium hydrogen exchanger. So hydrogen gradient here, the downhill hydrogen gradient is created and powered by a downhill sodium gradient. So if you poison the sodium potassium ATPase here by like wabane or some other poisons that inhibit this, it not only affects this, but will also ultimately affect a proton-dependent transporter here. So this is proton-dependent, and it's powered by this proton gradient that is generated by the sodium potassium ATPase. So once these are di and tripeptides are found in the cytosol, they are attacked by cytosolic peptidases, dipeptidases, tripeptidases. The basolateral membrane has no di and tripeptide transporter. It only has a platoon of basolateral amino acid, transporters. So here you have only about, again, 6, 7 free amino acid transporters, bringing amino acids from the cytosol to the blood side, where we partake of them because we need proteins for structural proteins in the body. Okay, so that would be it for today. For on Friday, there would be an online review. And for those of you who are online, please send me your questions by email, and attached on those emails or embedded in those emails are the specific slides. So thank you very much for attending online or in person, and thank you. I need to stop recording.